Okay, hi Dr. Lin. Today my group is going to present to you our topic on estrogen and insulin sensitivity. I'm Wilson and these are my group members. Okay, basically for estrogen, right, there are various types of estrogen like estrone, estradiol and estrotriol. They are produced from different organs, and for our topic we'll mainly focus on estradiol, which is the E2. Okay, for insulin, as we all know, this is the hormone for regulating our blood glucose level, and it's produced by the beta cells, the pancreatic islets. So a few functions are like glycogenesis, gluconeogenesis, and anabolic processes via the fatty acid and amino acids. Okay, as to how insulin helps to uptake this glucose is via the GLUT4 glucose transporters. So when insulin binds to the receptors, GLUT4 transporters will be translocated to the plasma membrane. So you will have a conformational change. So glucose will be uptaken into the cell to regulate the blood glucose level. Okay, insulin sensitivity. Okay, this term it kind of breeds the sensitivity of the cells towards uptaking this glucose level into the cell. So for normal human being, right, with no diseases like diabetes, so they will have a normal effects in the sensitivity level as to taking in the glucose into the cell. So a method that's used in the studies that we have done is like euglycemic and hyperinsulinemic glucose clamp. Okay, on the other hand, right, insulin resistance means that the cell is less sensitivity towards the glucose uptaking. So this happens during when at normal conditions. Okay, we also look into an estrogen and the estrogen receptors relationship. Okay, we have found that estrogen, the E2, actually represses this GLUT4 expression. So the body has a lower level of insulin sensitivity. And however, insulin resistance is also induced by lack of E2. So we can conclude from this that there's a correct level of E2 for a sufficient level of GLUT4 expression and a sufficient level of insulin secretion at the same time to help regulate the blood glucose level. Okay, a few mechanisms of estrogen on the cell in regulating the gene expression is by the membrane target firstly. Okay, the receptors is on the plasma membrane, so the estrogen will bind to the receptors on the plasma membrane and activate this downstream intracellular messengers like the other, in addition also to the kinases. Okay, for the cytosolic targets, right, the estrogen, because this is a lipophilic, so it will diffuse across the plasma membrane and bind to the estrogen receptors. So it will also activate the similar pathway as compared to the membrane target pathway. Okay, for the third one, right, the nuclear target, the estrogen will directly diffuse through the nuclear membrane and bind to the estrogen receptors, which is the alpha or beta that we have done also from our studies in directly regulating this gene expression. Okay, now we go to the discussion of findings. Okay, there are three types of receptors that we have done from our studies. Okay, the first one is the alpha receptors, as you can see. Okay, when there's a presence of E2, there's no increase in the insulin expression as compared to the wild type. As for the beta and the G protein estrogen receptors, knockout mice, there is a similar increase in the insulin expression. Okay, this sort of confirms the alpha receptors' role in regulating insulin's expression. Okay, on the other hand, there could be no significant levels of increase in insulin levels in the wild type and the ER knockout mice. Okay, this could be due to a difference in the cell location and physiological conditions in the cell. Okay, insulin secretion can be also independent of the estrogen receptors. Okay, when the cells are administered with glucose, there's an increase in the insulin expression in both the female and the male mice. This is seen also in both the knockout and the wild type mice. Okay, now I'll pass my time back to Freddie to continue with the rest of the results.
As you can see from this graph, the main takeaway from this graph is that there is no significant difference between the male and female mice in terms of the knockout versus the wild type. However, when the male mice or female mice has estrogen receptor knockout, the blood glucose level is higher than that of wild type. This goes to show that the estrogen receptor is important in regulating blood glucose. Also from our studies, we have found that the whole body insulin sensitivity is significantly higher in the wild type than in the alpha estrogen receptor knockout mice. Okay. There are also other effects of estrogen and estrogen receptor, such as adipose tissue regulation, energy expenditure, protection of the islets of Langerhans from apoptosis. This graph shows that there's increase in adipose weight for the knockout mice than the wild type mice. This is because since the estrogen receptor is the one regulating the conversion of insulin to glycogen, and because there's this receptor is being knocked out in the knockout mice, the shift from glucose is now converted to adipose tissue. As such, the tissue weight is now increased in some of the organs such as the kidney. This graph also shows that the body weight is significantly higher in the knockout mice than in the wild type, and also the food intake is increases in knockout than the wild type. The prevention of apoptosis of beta cells is done through the increase of calcium ions by E2, and this results in the rapid activation of chem response element binding protein, which is a key transcription factor involved in beta cell division and survival. The estrogen receptors are also involved in incrementing insulin biosynthesis and GSIS, thus reducing apoptosis rate. Okay. We have found some strengths and weaknesses in our findings. By studying mice, the influence of estrogen and its receptor to insulin insensitivity can be established. However, the knockout mice may produce different characteristics from humans in which the same gene is inactivated. As such, further research needs to be done to see if the same results can be replicated among human models, and through this we can obtain medical value from these experiments, such as gene therapy among diabetics. One of the studies is that we are able to find other factors and genes responsible for insulin sensitivity in other organs like our liver, which also governs glucose homeostasis. This, of course, has potential medical purpose in gene therapy for type 2 diabetic patients. However, more research needs to be done on the function of estrogen in various organs for insulin sensitivity. Current research trend also shows that the shift from estrogen receptor alpha to beta, hence there is now more focus on the beta estrogen receptors. Thank you for your time. These are our references.